So we have a number of co-authors and also some guests invited to participate in this presentation. And my idea here is to give you an overview about the progress of this global paper about fungal endemicity and vulnerability to global change. So I'm trying to share the screen now. I have too many windows here. Okay, I hope you can see my screen now. This project is funded by many different sources, Estonian sources, European sources, and also sources from various collaborators. I also ask you to mute your microphones because I hear a lot of noise coming from other people. So you are most welcome to write to me if you have any specific questions or ideas about the presentation. If you want to communicate this personally, then it's okay. But we also have plenty of time after this presentation for public questions. So let's start. Our objectives of this large study were to address the endemicity of fungi on a global scale and also to study the vulnerability to soil fungi to various global change predictors, mostly drought stress, heat stress, and also land use change. The idea is also to update global biogeographical patterns of fungi and determine the predictors of alpha diversity and gamma diversity. In the forthcoming manuscript, of course, we are also comparing all these patterns to those of microorganisms and other soil biota, well, as little as these are available so far. And we also consider specific groups of macrofungi that have been considered particularly protection worthy versus other fungi, which also include pathogens, yeasts, and other not so well appreciated fungal groups. So here in the main slide, you can also see the main map of sampling with different symbols. There are samples from 5 different sampling designs. To go further, these 5 datasets include the global soil microbiome dataset that came out a few weeks ago. It includes 3,200 plots, into which a vast majority of you contributed to in terms of sampling. Thank you very much. Now you are a part of this team. In addition, we also have hundreds of samples from teams of Manuel Delgado Paquerizo, Fernando Maestre, Martin Zobel, and Ülo Mander. They all have a bit different sampling schemes, so inclusion of all of these was not that straightforward, and from certain analysis, we had to exclude one or more of these data sets. What is very characteristic of all these data is that we obtained the original DNA samples and perform resequencing with our own primers to cover the full-length ITS region and also small part of small subunit using PacBio third-generation sequencing, which is supposed to be very high quality and very high accuracy compared to short reads for fungal metabarcoding analysis. These methods are not very high throughput, so we obtained more than 60 million raw reads. We did quite thorough quality filtering, so that more than half of the reads were actually lost because of the stringent thresholds. We defined OTUs, or in the paper refer these to as species, at 98% sequence similarity thresholds. And after these steps, we also performed extra quality filtering to assess the validity of singletons, to remove index switch artifacts and potentially contaminated samples. On top of all these high throughput sequencing data, we also added Sanger sequencing data from GeneBank and Unite, only focusing on fungi from soil. And these include about 180,000 sequences from a bit more than 8,000 localities. So you see the contribution of this is mere 1%. And based on a fungal traits database, we assigned all fungi into 8 main functional groups, and we disregarded smaller groups for simplicity. I'm presenting the methods and results in the order they are going to be in the manuscript. So the particular attention is on endemicity, on vulnerability, and less that on general diversity, because this is largely covered in previous studies. So for endemicity, we selected Woodward's, Olson's ecoregions, which look like in this map. There are around 700 or 800 of these, so we had to aggregate the data to cover these nearly equally or reasonably. So on the maps in the right, you see that we had to take into account the floristic and climatic similarity of these ecoregions to sum these up into so-called super ecoregions, also accounting for the sampling depth in these original ecoregions. And in some cases, as you see below, there is a map of Estonia and northern Latvia, which are particularly oversampled. We actually had to reduce the numbers of samples taken from these areas to prevent overemphasizing these habitats. And we also considered countries such as Estonia and Latvia, and sometimes also some geographically distinct islands as specific super ecoregions. So each super ecoregion included from 1 to 40 GSMC plot, but it may have included extra plots from these 4 other sampling designs, and also extra data from Sanger sequencing. We also calculated the maximum geographic distance for each species occurrence, which is necessary for calculating certain endemicity measures. We found that around 50% of all species occurred just in one plot. We included these as well, and we assigned a constant minimum value of 100 kilometer geographic distance breadth for these so-called singletons. So terrestrial ecoregions again. And now this graph shows the super ecoregions, which have been then generated based on the original ecoregions. I'm sorry that colors of nearby ecoregions are often matching, because for this graph we first sorted super ecoregions by name, which was a mistake, so we have to redo it. So all in all, there are 177 super ecoregions, and in the paper we will actually refer these to ecoregions to avoid this word super in every here and there. For endemicity in particular, we generated a super index. We can't avoid super here. It consists of 5 different indexes. First, it has a number of endemic species, percent of endemic species, then 2 similarity indexes, which are based on the area, beta Jacquard and beta Sim, and also a very different endemicity index called CRISPS weighted endemicity. We combined all these indexes by summing 1/3 of contribution of 3 of those index groups, and we took into account the sequencing depth and sampling depth, and Z transformed these variables for comparison. For vulnerability indexes, we tried quite a few, but obviously there was a simple best index which conformed to our expectations. This was T2 or V2 area-based index of Smith et al., published a year ago for lichens, in fact. For vulnerability, we calculated T2 vulnerability scores according to maximum monthly temperature, minimum precipitation in the driest quarter. So these most strongly represent heat stress and drought stress, respectively. We also considered land use change, and we are having a quite hard time including also pH change. The main problem here is that the effect is strongly unimodal, and the database values we have to rely on are very imprecise and calculated based on water, whereas our original values are calculated based on potassium chloride. We are also calculating or recalculating averaged vulnerability, and here the main problem is how to handle potentially overlapping or synergistic effects between those different variables. And in these calculations, we use the year 2015 as baseline, and the year 2040 as the extrapolation year. And we are using the climate change scenario 8.5, which means going on business as usual with marginal actual reductions in the greenhouse gas emissions and land use changes. Fortunately, there was no sequencing depth effect in vulnerability, and we also performed modeling of these various vulnerability predictors using random forest and GLM. For alpha, beta, and gamma diversity, we compared plot level and ecoregion level approaches. We also included land use interactions with various variables, and also included quadratic terms in the models. For latitudinal gradient revising, we simply used correlation with frigidness to the distance from the equator. Now to principal results. Our analysis in total revealed around 2 million eukaryote OTUs, of which around 960,000 belong to fungi. Here below there are enlisted those principal functional fungal groups, including non-mycorrhizal agaricomycetes, or better say non-ectomycorrhizal agaricomycetes. These are mostly decay fungi, a lot of polypores, a lot of stipitate decomposers. Ectomycorrhizal fungi, arbuscular mycorrhizal fungi, molds, pathogens, opportunistic human parasites, unicellular, early diverging fungal groups, and yeast. So these analyses revealed that perhaps the genus Tomentella is the most species-rich group globally, followed by Penicillium, Mortierella, Russula, Cladophillophora, and only then Cortinarius and other groups. For some groups, of course, the species may be a bit overestimated, whereas for the others it's underestimated, because we know that resolution at the level of species may differ strongly between, for example, Penicillium and Clomus. In Penicillium, there may be a single base pair to separate among species, whereas in Clomus, the intraspore variability may be the range of 2% or even more. This global soil microbiome consortium dataset was the largest of these databases. In itself, it comprises 3,200 plots and more than 700,000 fungal OTUs or species. All these had very strict standardized sampling design. Samples included known metadata about vegetation, different soil properties, and estimates about biome and vegetation age, etc. When this is compared to another large data set, global fungi, we see that the GSMC full ITS data set has much larger proportion of non-basidiomycete, non-ascomycete, or non-dicaria phyla, whereas global fungi ITS2 dataset has only a very small fraction of these. So there may be strong primer biases and strong identification biases. We used very heavily the UNITE database. For this particular project, we generated a series of new versions of reference databases to include more information about the origin of the fungi, especially focusing on soil and also on the validity of taxonomic annotations. Much of this was done actually through generation of the fungal traits database. This was another integral component of this global study, especially for functional annotation of fungal groups. So principally, this fungal traits database is the update of our last paper, 2014 in Science, and also fungal guild trait database. It has now 17 lifestyle related traits, including overall guilds, biotrophic interaction capacities, the composition capacity, fruit body and hymenium type, growth form, aquatic habitat, interacting taxa, if known, and some additional traits for ectomycorrhizal and lichenized fungi. The other part of these annotations was performed at the level of species, at the level of sequences, and these were later added to the UNITE species hypothesis. So coming back to the methods and results, then for endemicity patterns, the fungal taxonomic and fungal guilds differed quite substantially in all different diversity parameters and also endemicity patterns. But it was good news that the endemicity indexes were quite consistent within fungal groups. Endemicity scores or values were mainly driven by the average mean annual temperature, average latitude, average precipitation or pH, but not by their ranges, ecoregion size, or isolation. By isolation, we mean the island habitat and the island distance from mainland. The only island patterns were evident in avascular mycorrhizal fungi, but these were quite weak. So the first figure to be presented also in the manuscript is the overall fungal endemicity. We are lacking a scale from this figure. So the dark green represents very high endemicity, and dark blue represents lowest endemicity, with all these additional values in between, and yellow indicating average endemicity. So for all fungi, you can see that all the ecoregions around the Amazon basin are very highly endemic rich for fungi, and there are a few other tropical smaller hotspots in the coast of Central Africa, Sri Lanka, and surprisingly also the Yucatan Peninsula. Yucatan Peninsula differs from others by that it's mostly dominated by dry tropical forests rather than moist forests. There is quite a long dry season, and also the soils are quite neutral compared to other tropical soils. And here, average mean annual temperature was the best predictor for fungal endemicity. And you see this slightly U-shaped pattern with the highest temperature values tending to have highest endemicity. Well, which principally confirms these results. To directly compare to other macroorganisms in terms of endemicity, first let's take plants. As you see, also plant endemicity is concentrated in tropical areas, but very heavily on larger islands, also smaller islands, but in terms of total number of endemics, the smaller islands have lower number of endemics, although the percent may be higher. But these same particular regions we highlighted in the fungal map are not so super high in plants. When it comes to vertebrate animals, several groups taken together, these patterns are again very similar to plant patterns, but the difference between tropical and temperate regions is much stronger. And again, typically those super high endemism centers do not match those in fungi. Well, of course, we need to consider these differences carefully, because all the methods from observations to ecoregion sizes and analytical stages are very strongly different between fungal and these macroorganism approaches. In this figure, you can see the main predictors of endemicity values for all fungi across those 177 super ecoregions. Besides this U-shaped effect to temperature, endemicity steadily increases with average mean annual temperature. It decreases by distance from the equator. Yeah, actually, I have to apologize. I messed it up with another figure. So this is showing the different components of all fungal endemicity. So the number of endemic species, this is most strongly explained by mean annual temperature. The percent of total endemic species, or the relative endemicity, it's negatively explained by distance from the equator. The mean maximum range of species has U-shaped relationship with absolute latitude. Jacquard diversity decreases with absolute latitude, and beta sim dissimilarity index increases with total latitude, indicating that the highest values are in the southern hemisphere and lowest values in northern hemispheres, temperate and boreal habitats. Here are the corresponding maps of other major fungal functional groups. For the manuscript, these go to supplementary material and including the best predictor as an inset. So the major patterns are the following. First, non-mycorrhizal agaricomycetes follow the pattern of all fungi. Avascular mycorrhizal fungi are showing highest endemicity also in tropical areas, not only wet tropics, but also in dry areas such as Arabic Peninsula, India. But the top area for avascular mycorrhizal fungi is Yucatan Peninsula. Not sure why. For ectomycorrhizal fungi, there are several hotspots, mostly in the southern hemisphere. For example, New Caledonia, much of Australia, New Guinea, and also the very southernmost South America with Nophophagous habitat. For molds, again, surprisingly high diversity of endemics in the Amazon basin, but also in the farthermost Russian Far East. The opportunistic opportunistic human parasites follow the same patterns of molds, well, partly because the order Mucorales, or specifically the genus Mucor, was overlapping between these two groups. For overall pathogens, again, mostly tropical habitats here and there, with no very clear patterns. The same also for various early diverging unicellular organisms. But again, you see this Yucatan Peninsula as outlying with a high level of endemicity. And yeasts also with no very strong patterns. So these are the best predictors of each fungal functional group. For example, non-ectomycorrhizal agaricomycetes endemicity is declining by distance from the equator. Ectomycorrhizal fungal endemicity steadily grows with the mean annual precipitation. Opportunistic fungal pathogens, opportunistic animal or human pathogens, endemicity tends to decline with the soil pH. For unicellular fungi, the decline is in the gradient of latitudinal gradient. For vascular mycorrhizal fungi, the pattern is strongest with distinctly highest endemicity patterns at the warmest ecoregions. For others, molds, pathogens, yeasts, the pattern, these gradients in endemicity are somewhat weaker. So these will be again added as insets to the specific maps in supplementary materials. We also add to supplementary materials the tables with all model predictors for these groups with specific predictors for various endemicity groups for all fungi and for fungal functional groups. We include only the average endemicity predictors of this super index. Second, vulnerability. The vulnerability figures are not yet complete for technical reasons. We are currently actually moving between different buildings, so there have been some delays in this analysis. But anyway, these are how these figures should look like. We are probably including the average vulnerability across all vulnerability scores for the scenario in 2040. This particular map is from vulnerability to heat, and this is best predicted also by this maximum monthly temperature, and in fact quite strongly so. So essentially it indicates that the ecoregions or areas that are already experiencing very high maximum temperatures are substantially more vulnerable to higher temperatures compared to habitats that are not that warm now. As supplementary materials, we include maps for other predictors and also the uncertainty maps for all of these predictors. For example, this map shows here that the measurement uncertainty is much higher in northern tundra habitats compared to most other regions. It's because much of this area is covered by ice, and we don't have, it is actually out of the temperature scale that we have here. This is quite an interesting map, which indicates the relative vulnerability of all fungi to drought, heat, and land use change. So different colors and their combinations indicate which type of stress is highest in particular regions, according to extrapolations by the year 240. This is intended to be figure 2B in the main manuscript. As I mentioned, vulnerability analysis are not fully complete, so we will include also at least 2 supplementary figures about the vulnerability of all fungi to each global change factor and average vulnerabilities of functional groups to global change, and also supplementary table about best predictors of these particular vulnerability values. So now coming to updates in global fungal richness. Again, unfortunately, these analyses are still in progress, partly because of technical reasons. So this particular figure is taken from the paper in 2014, but the initial maps actually look quite similar for the whole data set. So again, highest diversity in certain tropical regions and lowest diversities in desert areas and also in Antarctic. These are also shown in these graphs. As you see, the graph by full latitudinal gradient from south to north. The Antarctic and subantarctic samples have lowest diversity, but then there is no much difference between South American temperate rainforests and also boreal forests or Arctic tundra in Greenland or Svalbard. So these are relatively similar in statistical terms. The deserts with lowest annual precipitation are also distinctly with lowest fungal diversity. So the critical line is around 200 millimeters of annual precipitation per year. The strongest predictor of overall fungal richness was soil pH, and it had this strong and distinct unimodal pattern, particularly decline in richness at strongly alkaline soil. These strongly alkaline soils are actually present in deserts, so there may be some confounding effect between the desert habitat and soil pH. Fungal richness is also steadily increasing with age of vegetation. The lowest stage is present in croplands, of course, and some short-lived grasslands. And these 300 and more year-old habitats are almost exclusively undisturbed forests. When it comes to different biomes and habitat types, we see that subtropical forests have highest diversity in total number of species per site, but these are quickly followed by other types of forests, also woodlands, some grasslands, also croplands and urban areas, with slightly smaller richness in boreal forests, shrublands, and particularly in deserts. The other land use graph in the very right illustrates these patterns, but it also shows that broadleaf forests and particularly mixed forests have higher richness compared to coniferous forests. But again, this may be partly the boreal forest effect, because almost all boreal forests, except some in Norway, are actually conifer-dominated forests. From alpha diversity to gamma diversity, quite surprisingly, or perhaps not, the richness patterns of gamma diversity are better supported. We see more significant patterns, although the number of super ecoregions is almost 20 times less compared to the number of original plots. So we see that the gamma diversity of fungi cumulatively increases with average annual rainfall. It also increases for ectomycorrhizal fungi. Richness increases steadily with the total latitude. So it's almost always lower in the southern hemisphere, and then the major peak is in temperate forests, which is quite close to boreal forests and tundra ecoregions. For human parasites, the main effect also goes in the mean annual precipitation axis. For agaricomycetes, again, one of the strongest pattern along the annual precipitation. The same applies for apuscular mycorrhizal fungi and molds. And it appears, in contrast to other fungi, that yeasts, the richness of yeasts is particularly sensitive to very high maximum temperatures. So these graphs, these tables illustrate the best models for explaining richness, both gamma diversity and alpha diversity. Alpha diversity also includes Shannon indexes for all fungi and functional groups. Both of these are presented as supplementary materials. We also revisited the latitudinal gradient of diversity. The top panels represent alpha diversity, and bottom panels represent gamma diversity. Both are for supplementary materials, and also it is evident that the gamma diversity patterns are relatively stronger compared to alpha diversity latitudinal patterns. For example, in all fungi, the latitudinal gradient is virtually absent, but for gamma diversity, it is slightly but significantly decreasing, although the explained variation is about 5%. And again, for many other groups as well, the gamma diversity is either increasing with latitude, The only example here is actually ectomycorrhizal fungi, but for most other groups, gamma diversity is higher in tropical latitudes, with strongest pattern in avascular mycorrhizal fungi. Taxonomic composition at the level of species is relatively poorly explained in this analysis, but you can imagine generating this huge matrix with 99.9% of zero values. There is actually no chance to get very good results. So for these studies, we excluded rigorously species with less than 5 occurrences, or some cases even with less than 10 occurrences, to get any reasonable patterns. So what is evident here is that either mean annual temperature or soil pH prevail in explaining fungal composition in these general or generalized dissimilarity models graphs, which also account for non-linear patterns. Those two predictors, temperature and pH, these differ among different fungal groups. But the overall patterns are that climatic patterns tend to be roughly linear in the logarithmic scale, whereas soil nutrients tend to have relatively stronger effects when the nutrient concentrations are very high, or let's say in the top quantile of logarithm transformed nutrient concentrations. So there is no strong sensitivity to very low nutrient concentrations in any groups of fungi in terms of composition. These GDM models, they enable also to include geographic distance based on the coordinates, and we see that it almost always explains less than 1% of variation. Avascular mycorrhizal fungi surprisingly have one of the strongest geographical distance effects. In all analysis, we also included temporal eigenvectors. These should be interpreted as actually spatiotemporal vectors, because we collected samples from one area, usually in certain time. For example, we visited Madagascar only once, and within this 3-week time, we collected all 16 samples from Madagascar. And what comes to also to geographic distance, so the maximum autocorrelation detected was in the range of 350 kilometers, not more, when taking together both latitudinal and longitudinal variation. Again, for supplementary materials, we present the best models of composition based on Permanova, and based on these two analyses, it was evident that there are some conflicts. These conflicts may come from nonlinear effects, also different capacities of these models to include binary variables, and sensitivity to very low matrix connectance. So here you can see that maximum strong effects are about 2 to 3% of explained variation across all groups based on Permanova linear only effects. So to conclude from this study, fungi have quite strong patterns of endemicity, and the patterns in arbuscular mycorrhizal glomeromycota seem to be the strongest, but there are no good statistical ways to test this really. Various fungal groups have quite similar vulnerability to global change predictors, mostly to high temperature, and this is also shown in some laboratory pure culture tests that all fungi, irrespective of their yield, tend to be sensitive to high temperatures. And it was surprising to see that particularly yeasts in our data were specifically sensitive to temperature stress. In this study, we update also fungal alpha diversity patterns and add gamma diversity patterns. Based on this large data, we can conclude that deserts, and in particular, or particularly Antarctica, also have lowest diversity, and we also can speculate that certain very acidic wetlands have relatively low diversity. So these wetlands are mostly represented by bogs, but unfortunately, we don't have enough bogs samples in our data set. The latitudinal diversity gradient is mostly negative, and these new analyses also include gamma diversity and also phylogenetic diversity of fungi. Compared to other organisms, at least based on visual and some kind of numerical comparison, the endemicity of fungi is less than we can observe in macroorganisms, particularly in vertebrate animals. There is no way to compare the vulnerability of fungi to other macroorganisms because in the way our approach is novel for detecting these vulnerability patterns overall. The global diversity predictors all differ from both other soil biota and also macroorganisms. The latitudinal gradients also differs from other soil biota and patterns described for macroorganisms. So with this, I conclude my presentation, and I take chance to thank the founders, all contributors who provided samples, analyzed certain aspects of the data, and here listed are those persons who are the core team for sampling and analysis. I actually see that a few persons are missing here, Alex Siska and Alex Antonelli. So thank you all, and now I'm happy to take your questions either regarding the manuscript or analysis, and all suggestions are welcome either here now or later by email. And also in about 1 week, I will provide you a link to the draft of the manuscript, which then includes all illustrations as well in full or almost full format. So I will inform you when it's ready. So now your questions, please.
I can start, perhaps. Jan Bern from Tartu here. So when we highlight the temperature and precipitation as the main factors of endemicity, I was thinking, well, they're surely not the direct mechanisms. So I was thinking, well, yeah, temperature and moisture itself, I guess, doesn't create the endemicity. We don't say that, but well, they increase or they decrease vulnerability, they increase productivity, and also from previous work was known that they increase diversity. So there's like a complex of influences there. So how do we, I was thinking, what's the thought process or the reasoning behind the explanation?
Well, there are certainly very many processes behind. Yeah. And in old days, they used latitudinal gradient also to take together both temperature, precipitation, and all other climatic predictors.
Yeah.
So when we take, for example, mean annual precipitation, so it seems to be some kind of best proxy to soil moisture availability.
Yeah.
And,
but still, is the direct influence through productivity or diversity itself, or decreasing vulnerability?
All those processes that we can easily measure are not reflected by this mean annual precipitation or mean annual temperature. So there must be some kind of lower level processes or some kind of general conductivity, or then some kind of micro evolutionary processes that are enhanced by these figures. So we actually do not know the mechanisms, the direct mechanisms, and these are also extremely difficult to measure. Maybe we can take this coronavirus as an example, and well, as it is the best studied organisms in the world already, and try to make some kind of extrapolation based on this, as it infects humans or as it spreads in the petri dish.
Well, evolution is definitely one key to consider. For example, I started to think when I saw the Chukotka Peninsula highlighted in one of the analyses that, well, compared to regions on the same latitude, it has quite special history and more perhaps stable conditions or longer exposure from the ice sheet. So maybe some analysis that consider this evolution or simply stability or history, we could do some more of those, or include a few more like historical or stability parameters.
Yes, I agree, and it's also known from some fungal literature that this long-term climate stability, especially in the northern temperate habitats, plays some role in the distribution of truffles, for example, that are relatively more slow to spread because they require certain mammals for distribution.
Mm-hmm.
So we can, yes, try these additional models about stability as well.
Another thing that I noticed was, well, the coastal equatorial Africa, and not by itself, but maybe the fact that it was higher in endemicity than the rest of the equatorial Africa, which is a bit inexplicable for me. And maybe the same goes for Borneo. So maybe it's worth checking out. There are not so many samples from the rest of equatorial Africa or African rainforest or Borneo. So maybe it's worth checking out those especially. So maybe they represent some special cases like peat swamp forests with lower pH or other special cases.
Yes, well, when it comes to tropics, it seems that areas, let's say larger areas with highest precipitation seem to be the major endemicity hotspots. So it actually may matter if you have like 1,500 mil rainfall or 5,000 mil rainfall.
But those African rainforests, they get those top levels of precipitation, no?
It's only on the coast. So the central Congolian forest is not that wet, in fact.
Okay, got it. And
okay, Carlos has a hand, please.
Thank you, Leo. I have a question related to the how you consider or plan to consider to calculate uncertainty in space. So you presented a very high distribution of data points, but have you considered any measurement of how uncertain are your predictions in some regions? Because some regions have less data points, for example, or have none. Maybe we can do something there to at least to properly illustrate what parts of the world are covered in the same manner or not.
Well, it means that this uncertainty has to be done in several nested steps. Probably if we include only those sampling points, their distribution and their variability in the geographic space, it might be sufficient. But of course, well, we have to really think of it further, because it is important to show self-critically that it matters.
Sure. So so I can also help on the first one that you mentioned. So at least for me, it would be the very basic would be to demonstrate how what is the environmental coverage of your sampling points at the moment. This would be something that I can also support you on that. But then what you mentioned is also important. So for example, if when you're evaluating, for example, endemicity, if you are subsetting your data or if you are doing something to the data pool, then we can also do the same. So we can perform the same calculation on those specific subsets, for example. This is something that we can also do. And what I would suggest for that is that we consider only the variables that you are using for your calculations. So for example, like you're mentioning temperature, precipitation, others. So this pool of predictors that you are already considering, I would not consider more, but I would consider those to at least understand what is the environmental coverage that you have.
Okay. Yes, we need to think about it in more detail. Yes. So usually, I guess it's the last step where to build those uncertainty plots and such types of calculations. And we can also see what the reviewers think, because it's obviously impossible to do everything. So if we submit and then see what the referees think we are missing, then it can be quite easily be added. Otherwise, it's just too much work to add all those potential, take into account all those potential shortfalls. Okay, Kui Heng has hand. Kui Heng, please. Or Nadia has also hand. Nadia.
Hi, Leho. Hi, everybody. I have a question. You are paying a lot of attention to environmental drivers, but for endemicity, according to my understanding, the level of human disturbance and, well, level of isolation of the locations from the rest of the world, connectedness, you know, through water streams or through people trading or something, that's also probably a very important factor. I understand there was not enough space in this work to address it in detail, but how would you argue why you didn't do this? Or you are planning to add this aspect?
Well, we have in a way taken into account these variables. For example, for isolation, we have generated a separate binary value as island versus non-island. And also for islands, we have accounted for this distance to the nearest larger island or nearest continent. And what comes to human effect, we have taken into account in biome and land use, but unfortunately, we don't have this very site-specific data about whether there was some kind of rubbish or whether there was some cutting or forest fertilization or something like that. So we don't have these very site-specific details.
Yeah, I was just remembering your maps, which you were showing this very high levels of diversity and endemicity in Amazonia area, for instance, seem to me to be very likely to the fact that it's such a very unexplored and in a way disconnected area. I think I understand your reasoning, but I think it should be very smartly addressed in the manuscript, maybe posing the questions, the hypothesis which you test, so that this aspects become not relevant. Because otherwise it seems that very important drivers are addressed not fully enough. Just a concern.
Yes, well, thank you for this concern. We will have to see if we can find any such large global databases for more specific human impact other than those Copernicus land use maps. So we will see. Okay, Guiheng, are you now able to answer?
Hello, can you hear me?
Yes.
Okay, hello, Neil. My name is Guiheng. I'm from China, Peter Martinez Group.
Okay.
So my question is about the driver analysis. So you're talking about the driver for the endemicity or the richness. So what's the standard for you to choosing these drivers, like soil pH or temperature? Do you have include any other like a vegetation index, like a NDPI, NDVI, something like that?
To be honest with you, we included all those possible indexes that we could get from various databases in the first modeling exercise. And this first modeling exercise we did using random forest. Based on this random forest, we selected around 20 best predictors, and then we continued with general linear modeling and also added those quadratic terms and interactions with categorical predictors.
Okay,
so we essentially we take into consideration everything we could, and then we refined it to smaller sets and did more specific and detailed analysis using these.
Okay, so you use random forest like a pre-selection model, right?
Yes.
Okay, that's come to explain my second question. So I just wondering why don't you use some multiple factor models? So now I can see that. Okay, thank you, Neil. Very fascinating work.
Thank you. Any other questions, please?
Well, if I may, I would continue from Nadia's point, and well, in fact, what I had the same thoughts exactly, that especially explaining the high endemicity of the Amazon, especially compared to other tropical rainforests, maybe the temperature and precipitation are not enough still, and the Amazon, while being relatively natural or untouched region, it still the sites may be isolated in a way that we cannot well quantify at the moment. So I don't have a ready index to offer, but well, it's definitely worth consideration whether yeah, isolation by rivers or just by distance from other I don't know, areas, not just by water, but just by distance. It could be well built into a isolation index that would distinguish the Amazon from other similar regions with a lower endemicity.
Well, actually, we used this isolation, but we used it on islands, and on islands it really didn't work. And therefore, I suspect that if it didn't work on remote islands, such as New Caledonia, then it shouldn't work also in some kind of regions within continents. But I agree with you that one of the main effect may be that the Amazon area has been rainforest for millions of years in a row, although there have been some savanna intrusions and so on, but it is still much less compared to, for example, Africa, which in glacial maxima had only very small truly rainforest patches.
Okay, so that's basically a stability index that could be developed from that thought process.
Yes, and I think it's quite possible because there are such studies also available, especially with a focus on plants and some also with a focus on animals. It's especially for Africa. I'm not very sure how about globally or South America in particular.
Okay.
But on the other hand, it wouldn't explain the Yucatan because Yucatan has been underwater for much of the geological time. It's all lowland area.
But again, is Yucatan not very well isolated from other, at least similar rainforest areas?
Well, much of the southern Mexico is this kind of dry tropical forest biome. But what is specific about Yucatan, maybe that it is the soil is developed on coral cays. So it's all very limestone based. And this kind of neutral pH, which is unique in that area. It may be very important for this Yucatan in particular.
Well, another thought is to develop a uniqueness index, like a tree-based uniqueness, tree-based approach from the environmental characteristics.
Yes. Much of this uniqueness is actually within this measure of the number of endemic species and the proportion of endemic species.
No, I meant in environmental characteristics, leaving the fungal data out.
Okay, yes, that can be certainly achieved using some kind of PCA scores, I guess.
Yes, or just like a similarity tree. Or distance in the tree.
Yes. Well, it's certainly worth to think about it further. Thank you. Karin, please.
Hi, Leo. Thanks very much. This was a very stimulating presentation. If I remember correctly, the moles were particularly endemic in Amazonia, but also in the northern Russian areas. Is that right?
Yes, it came out from the analysis.
Yeah, I think this is puzzling. What are your thoughts on that?
Well, to be honest, I don't think it's so much true. So that there may be also some kind of additional, let's say, technical factors that may contribute to some of these patterns. Because, you know, molds, they are also sometimes considered as contaminants. Not direct contaminants, but these may come from, well, when you have spoiled samples. So they may have then very high abundance. And when they have very high abundance, there may always be some kind of artifactual sequences and all sorts of similar problems. So, and in these particular cases, these samples were quite rich also in molds in terms of relative sequence abundance. So in that particular case, it may be that this mold endemicity is overestimated in those particular areas.
Is this what you would respond to the reviewers then, or are there any other thoughts on how to present this data? Or, well, because this is, it somehow confounds the other results a bit to have reliable data. It's just how would you address this to avoid that people question the overall quality of the results, let's say?
Well, they can always question, and
sure.
they always do in case of my research.
No. Yeah, I know what you mean, but
there are always so many open ends when you do this kind of environmental descriptive studies. Then you can always select some different techniques, and people who are using different methods, they of course think that, okay, why didn't he do this way or that way? Well, the truth is somewhere in between anyway. So we are just trying to get as close as we can based on the methods we can use.
Yeah.
But yes, of course, when we respond to the reviewers and they ask these things, well, certainly we must look more closely to the actual data and see whether any of these technical issues might be the case. But this reply to you right now was based on the fact that I recall that there were several problematic samples from these particular areas.
Thanks a lot, Leo. But otherwise, super, super interesting work, and I really enjoyed it.
Yeah, thank you. Okay, any other comments, questions, please?
Can't find the exact reaction here.
Yes, Kadri.
Okay, so you have these global maps for both richness and endemicity. So except for this Yucatan, what do you consider as the most outstanding contrasts in both ways, like having low diversity and high endemicity in fungi, or the other way around? Have you compared or thought about that, in which way these things, whether they are correlated somehow?
Well, they certainly are, especially when we take those very specific endemicity stats. Then some of these are strongly correlated to richness. For example, the number of endemic species. Well, it's certainly correlated, but the proportion of endemic species, it's not correlated at all. And that's why we use this kind of super index to iron out most of the wrinkles in those different endemicity measures.
Yeah, I definitely ruled out not to have this correlation, but now are there any other outstanding areas? I noticed, for example, it seemed to me that Australia didn't have very high endemicity, like compared what we know about plants and animals. So I would assume that there are many, because the plants, there are a lot of endemics and there are many plant-associated mutualists and parasites. So why do you think, what is the reason? Is that the global dispersal of spores, or why this effect in Australia, and as was outlined by others also in several other islands, the effect of, or the indication of endemicity isn't very high?
Well, certainly much of these patterns are driven by those so-called microfungi that disperse very efficiently across continents. The other reason may be that using the current identification methods, we really cannot determine some of those microfungal groups at the level of species. For example, the cases with Penicillium, Fusarium, Alternaria, etc. So these are still certainly some kind of species complexes. And if we could go into more detail in these groups, we might see much stronger patterns of endemicity. So it's probably the similar case to arbusculum mycorrhizal glomeromycota, because the previous study by John Davison, it seems to be more on this kind of super species level or subgenus level patterns based on the small subunit, whereas in this particular study, those 4,000 plus glomus species alone, these are certainly somehow, let's say, not subspecies level, but some kind of intraspecific level.
Yeah,
say some kind of genotype level in that case.
So it might be one idea for a later sub-project to focus on some groups, like call them molds or some ascomycete, that to use the lower level of resolution and then see the endemicity pattern over again. I would still assume that these maybe almost cosmopolitan fungi cannot be so adopting to these endemic plants like in Australia and elsewhere. But yeah, thank you.
Yes, well, it was actually one of the main ideas. Also, people had chance to register to be able to, or to like, if they want to analyze some specific taxonomic group or continent or whatever. And the idea behind these taxonomic groups is just to select the best relevant taxonomic threshold for each group. So in case of Fusarium, it might be just one base difference in the ITS sequence, which would be something like 100% sequence similarity. And I assume that the patterns are generally similar, but absolute values of endemicity or other measures would differ quite a lot. Okay, any other questions? I can't see also in the chat. There is question, how many samples were collected in Yucatan? I think there were around 15 samples from this super ecoregion. Okay, thank you for everybody attending. In around 1 week, I will send you, I will send the co-authors specifically the link to the manuscript. And meanwhile, I also will discuss with people's presenting some ideas or suggestions, and how to most reasonably approach these questions. So nice to have you all here, and enjoy the rest of the day. Bye.
Bye-bye, thanks.